Thanks everybody for joining us. I'm really pleased today to be able to introduce Dr. Peter Marks, the director of the Center for Biologics Evaluation and Research at the FDA. Peter doesn't need much introduction to this community because he's been a really important part of our world over the past several years. He comes to this role with deep academic background, a career in the pharmaceutical industry, and a really distinguished tenure at the agency. He's been a terrific partner to ARM in our community, and I'm really pleased to have a chance to talk to him today about the current state of cell and gene therapy. We're going to start by giving Peter a chance to make some opening remarks about the state of cell and gene therapy and the agency's work for 15 or 20 minutes. And then when he's had a chance to do that, we'll come back and have a Q&A. Thanks again, Peter, for joining us, and I'll turn it over to you.
So thank you, Janet. Thanks very much for having me today. What I'll try to do in a couple minutes here is kind of take us on a brief tour of what we're working on at the agency and some of our areas of interest and where we hope to be heading. So from the standpoint of where we are now, we're in a state where we have several approved gene therapies now, either cell-based gene therapies or directly administered gene therapies. And there really are, as I'll show you in a moment, many cell-based and directly administered gene therapies in various stages of development. It's clear that COVID-19 has adversely affected all aspects of the development of cell and gene therapies to differing extents for different products. I mean, I think there's been some good news here, and that we still see quite a number of INDs coming into the agency, but it's also clear that things are not moving as fast as they were, and we'll say a few words about that. And it's also clear that manufacturing is remaining a rate-limiting factor in the production of gene therapies. So we're all aware, I think, of the gene therapy for spinal muscular atrophy. This systemic administered gene therapy is one of the, I think, one of the bright lights here in terms of what can be accomplished. Anasemnogene for spinal muscular atrophy type 1 really has a transformative effect. We have children that just previously would have clearly been not alive, who are, for all intents and purposes, normal at 3 or 4 years of age. And that made a clinical trial, that type of result, when you have it, makes a clinical trial relatively easy to do because you have an outcome that is simply not expected at a given point in time. So a single-arm trial works well. And the child on the right in this picture, this was a child who was brought to the Senate on Capitol Hill, and just quite a remarkable outcome. So this is what's possible. And the question is, how can we have this not be just a one-off or a 2Z or a 3Z? How can this become more the norm? So there's clearly a tremendous amount of activity here. And I'm happy to say that, you know, there were, if you look on this, on the slide here, towards the top, you see this graphic, which says that in 2017, we had 106 gene therapy applications. That doubled to 2018. It was over 250 in 2019. And we are well on our way, even in the setting of COVID-19, to exceed that this year in 2020. So I think this is a very exciting time in gene therapy still, even in the setting of COVID-19, and even with those challenges. It's just that for some of the studies that are ongoing, there are some real challenges to overcome in terms of endpoints that may have been missed, et cetera. Hopefully, we'll be back on track, so we will see the types of numbers of product launches that were predicted by the MIT group, with 40 to 60 predicted by 2030. It is, you know, it is, with that number of INDs, it's hopeful that we'll start to see products coming through. So, you know, now as of 2020, we have the 5 therapies, as I said, 3 of these being CAR T cells, and the two directly administered gene therapies, one into the eye and one systemically. And hopefully that will that number will increase nicely over the coming years. But what are the challenges here? So I think it keeps coming back to me to manufacturing often as some of the real challenges here, because we still don't have process scale up that's reproducible. From clinical trial lots to commercial lots, people move process from one contract manufacturer to another, and they're not even sure whether they've got the same activity. And that can be a challenge here, because in small populations, there's going to be variability anyway. And if you essentially multiply that by uncertainty in the manufacturing process, that can get challenging. I think some of this too is that because the manufacturing has been so challenging, manufacturing has been become something that's become very proprietary. How you make the secret sauce that you use to make your adeno-associated viral vector for administration, that's become a very proprietary. And so instead of having a dynamic exchange of information, there's much less dynamic exchange, and that means that I don't think the field might be as advancing as quickly as it could. And I think the cost here that results in is also impeding progress somewhat. Where do we need to go? We really need to have quality manufacturing of advanced therapies. That needs to be considered from the outset. In particular, I think for some of the gene therapies where you're dealing with a disease for which there is a dramatic outcome, like spinal muscular atrophy, and where you don't need a lot of patients, you have to be set up from the beginning to be successful, because if you have to transfer a manufacturing process, that translates into lost time and lost opportunities for patients to benefit. The high quality manufacturing capacity, really, we need to be able to have more of it. And we probably also need to be able to think about how we can streamline getting to that kind of advanced manufacturing. So this is a slide I've shown before, which says that, to me, right now we're in this sweet spot of manufacturing gene therapies at a scale which we can imagine administering from 100 to a few thousand patients with our current manufacturing technologies. If we're going to move up and actually have gene therapy address larger populations, particularly if it's not going to be locally administered, if it's going to be systemically administered, we're going to have quantum improvements in the vector technology and the manufacturing technology. And so that is something which I think will be really exciting to see happen. On the other hand, there are literally hundreds of diseases that could be addressed with gene therapy now, if we could figure out a sustainable way to make those gene therapies in terms of cost and production. And so that's why for us at FDA, we've really focused somewhat on this small cup where it would be nice to find ways to make it possible to have these therapies for those with diseases that are essentially devastating. I mean, there are any number of diseases which are devastating in either very early childhood or as children grow a bit, and it would be nice to be able to address those. So these very rare disorders that may only affect even a few dozen patients a year, there is this potential to address them with novel therapies, and gene therapy is a real way of getting them. Or some cases, it could be a cell-based gene therapy. But I would put it to you is that some would say, well, why would you really care about these small numbers? Why shouldn't we just be going after larger numbers of patients that you could benefit more? Well, first of all, it's because the trials for some of these where you have very unambiguous outcomes should be relatively straightforward. But also because I think if we can actually do this for many different small populations, very small populations over and over again, we're going to start to learn a lot more about critical quality attributes, key manufacturing parameters, and that exercise will actually benefit the entire gene therapy field. And it will be good, I would put it to you, for the larger groups of patients. We will benefit here. And this goes to this concept here that I think we are indeed moving from personalized medicine, where you find the right drug on the shelf to treat the patient, to individualized medicine, where we create the right drug to treat the patient. And if we're going to do that, we're going to have to be able to do it in a facile manner. There are some cases like customized products where we have an idea how we do that already, such as personalized cancer vaccines, how you might go about making a product that has the same indication and the same mode of action, but just slightly different characteristics that tailor it to a given person. But what we're talking about here mainly for these rare diseases are created products where there'll be different indications, different mode of actions, even if they might be based off of the same vector. Examples would be gene therapies for 2 different hemoglobin mutations where you use the same vector backbone. And because we're on this visual platform, we might as well think about it this way, that we really have this concept of personalized medicine, perhaps, where you had ready to wear, to this kind of customized medicine where you have made to measure. The suit's already there, but the seams are left open so you can get it right, to bespoke or individualized tailoring, where really we're creating something for an individual or a small group of people. And it's not so far off crazy, because when you think about it, even some larger gene therapy applications might break down into almost bespoke ones when you think about dealing with the genome editing of individual mutations that might be present. So where do we go with challenges of individualized therapies? We have manufacturing, which I said first and foremost. There are also obviously some others. Non-clinical development can be a challenge. Clinical development is still not a cakewalk, and product access. For non-clinical development, we know that animal models may be less than ideal. When you think about it, especially as we move to genome editing, using anything other than something that's human doesn't really help you a whole lot in terms of off-target effects, because unless that animal model has human genome in them, you're not actually going to find out what's really relevant for off-target effects. So can we start to think about model systems, organoids, humanized mice to help facilitate safety testing? In terms of clinical development, we really have to start thinking about how do we really efficiently determine efficacy in very small populations, because that's right now can be challenging. We collect baseline data. How do we deal with this for diseases where the natural history of disease isn't well documented? And can we start to think about novel statistical methods that could potentially expedite product development, such as using Bayesian designs, and potentially helping to just start by saying with each patient, you move closer towards getting across the finish line. And then for product access, how do we go about making sure that for these diseases that are uncommon, that we actually have a sustainable model, so that it's not just, a clinical trial that you do that provides the product, but that there's a long-term provision of product? And is that something that would be done by a public-private partnership, or could a public-private partnership even be something that transitions ultimately into something that could be commercially viable in terms of a larger ecosystem, where if the costs came down sufficiently, you'd have industry more interested in addressing these small populations. One of the things that the Foundation for NIH and the National Center for Advancing Translational Science has been working with FDA on is this Bespoke Gene Therapy Consortium. It's a project that's now been endorsed. And the idea here is to try to have kind of a nonprofit umbrella organization that goes from trying to address some specific rare genetic diseases, taking them through using AAV and leveraging using the same processes to ultimately bring a gene therapy along in a sustainable manner. This is kind of trying to use the razor blades analogy of understanding what a given AAV vector can do when made by a consistent manufacturing process, and then swapping in different inserts. The idea here is to leverage master files, leverage manufacturing technologies that are standardized, to see if we can try to bring down the cost, and to see if we can come up with a cookbook. It's like the molecular biology Maniatis for that analogy. This is the analogy of this for gene therapy, a kind of a cookbook to help these things along and to try to get them to get there efficiently. So we'll continue to use our expedited programs here to advance gene therapies. We're going to continue to publish guidance documents, meet with sponsors. We want to advance manufacturing technology, and we're doing that through a number of grants that we're able to give. We'll also continue to develop new regulatory policy. Our guidance documents now in the area of gene therapy, our suite of 6 guidance documents that we issued, are now all final. We recently issued an additional guidance on orphan sameness. This is trying to understand whether different gene therapy vectors are either the same or different, depending on whether they have the same insert, different inserts, same vector, different vectors. And one of the issues here that's come up is you can be a lumper or a splitter. The advantage of lumping, obviously, is, well, there are fewer things around, and it's easy to say that this particular gene therapy for disease X there are a few of them. It may give one entity kind of a leg up. The advantage to splitting is that you potentially allow for a lot of innovation upon what you can do for a given disease X, because you can imagine that you could have a first generation disease X treatment that is okay, but if you tweak that using that same gene by using some different regulatory element, you could have a better therapy. And so the idea here is that we've tried to make it clear that obviously if you have 2 different transgenes, they're 2 different gene therapies. If you have 2 different vectors that are clearly different vectors, they're 2 different gene therapies. If you have a same vector and the gene in it, but different promoters, different regulatory elements, maybe those could be different. If it's not, I mean, if it's just trivial, they might not be different. So it depends. But this, we hope, will stimulate innovation here. We continue to work with sponsors on our Interact programs, and we're trying to work towards improving the time delays for people to get to those meetings, particularly in this time of COVID. I am not at all proud of our ability to have these in a timely enough manner, but we're going to work on this. We have the platform meetings, these CBER Advanced Technology Team meetings for people who have for instance, maybe they have a vector platform or manufacturing platform they want to talk to us about. These meetings are available, and we're very happy to have those with sponsors. And then I'll just finish up by saying that kind of the policy concepts that we have in development are really this concept that I mentioned already for individualized or bespoke therapies, this idea of leveraging preclinical and manufacturing information from one application to another, and this concept of having originator products and offshoot products where you leverage information on file and really focus on the distinguishing attributes of the offshoot or the variant product. And then the other idea here that we're working with our statisticians on is this idea of this kind of continuous reassessment model in clinical development. This idea of trying to use Bayesian designs to determine, well, if you're only going to have one clinical trial, is there a way to figure out a boundary which, when you cross that, you have the equivalent of an efficacious product? So those are some things. We don't have all of the details worked out, but they're what we're working on. So just to finish up here, we're very committed to moving ahead gene therapies, cell-based gene therapies, and other novel therapies to help meet the needs of diverse groups of people with disease. And we'll do that through what we can to individualize product development, help with innovative clinical trial designs, work on novel endpoints, and really my sincere hope is that we can hope to overcome some limitations in manufacturing. Really, this is an area that's very, very exciting. It's what actually helps me get through my COVID-19 laden days, because this keeps me excited on the other end of getting back to moving ahead gene therapies. So I'll stop there and look forward to the discussion.
Well, thank you for that, Peter. That overview is really helpful. So you mentioned at the end that you enjoy getting to talk about gene therapy, because obviously there are a lot of other things going on in your world these days. So let's start there. Can you talk about, first of all, how you're doing in the midst of everything that's swirling around you? And really, given the demands that COVID is placing on the agency and on you, what is the split of your time these days between COVID-related work and, say, gene and cell therapy?
Yeah, so it's a good question. This is a very challenging time, obviously. I have, one of the lucky things about my life is I've always kind of been a little bit regimented in going about things. So I always try to set aside some time for dealing with gene therapies, even when it's been crazy through this period of time, where we've been dealing with the therapeutics, convalescent plasma, polyclonal antibodies, and the vaccines, which right now obviously are very top of mind. So I've been able to make sure that I've spent some time dealing with and focusing on cell and gene therapies, not anywhere near as much as I would want. Because if I think back to November of 2019, I would say I was spending 75% of my time, perhaps, on issues with cell and gene therapies. Vaccines kind of took care of themselves, and the blood products are something that I'm very familiar with, and we were able to deal with relatively quickly for a lot of the issues. Not that they aren't important, but that it was just some things have less policy demands at this point in time. Now at this point in time, it's very much reversed, and it's probably 80% of my time on COVID-related activities, and probably 20% on cell and gene therapies because of the nature of what we've had. That doesn't mean that I don't care very deeply about them, and it keeps me up at night worrying about what's going to happen when we finally see the light of day when COVID clears. I'm worried about the phase 3 clinical trials or pivotal trials that people have in gene therapies that may not be going optimally. I worry about the delays we've had in moving ahead with some of the advances in manufacturing. We were on the cusp, in fact, working with global regulators of trying to get towards more harmonization of the gene therapy programs in different countries. Because personally, that's a very important thing to me, because I view that our differences as regulators across the globe, it's actually very detrimental to patients, because it prevents innovation from permeating across the globe in the manner that it should. So if we could get our acts together to have, particularly for these small indications, to agree on what was necessary so that we could have almost more mutual reliance. It's not exactly the right term to use here, but to have mutual reliance on one another, we might be able to have more benefit to people. So all of that was on the cusp, but we're trying to keep that moving now, but it's a challenge to do. I'm hoping that as this winter, as we start to see vaccines come through, that we'll be able to get back towards focusing more on gene therapies and cell therapies.
Yeah. Well, you've raised a lot of issues there I'm going to come back around to, but before we kind of leave what's unique about the current moment, I wanted to ask you about the op-ed that you and a number of your career colleagues at FDA published in in USA Today, reaffirming your commitment to following the science and the good work that you do at the agency. I haven't seen anything similar to that in quite some time, and I've been in Washington a long time, so I can imagine, there was quite a lot of thought that went into a decision to publish something like that. And I wondered if you might just talk a little bit about that.
Yeah, what drove that, I think, is a very fundamental thing that I think is the public needs to understand, that we have an incredible workforce of people that are so committed. They come to work every day, or they put on their fuzzy slippers and go to their computer every day now, solely devoted to doing the right thing by public health. They drown out, they really do. They do their best to drown out the noise that's all around us right now in the political arena, which I'm not going to say much more about. But they focus on really the science here. They focus on caring about the people that they will ultimately help. And I know sometimes sponsors may think that we are heartless at FDA, but we actually do have hearts, and we really care about, people really care about what they're doing, and they care about doing the right thing. And what led us to do this is when we start to hear, don't mess with my family. You can mess with, you can mess with a lot of other things, but don't mess with my family. This is a group of people that we are together as one in being there to do what's right by the science, to make sure that what people see comes out of our agency, they can have trust in. And anything that impugns that is just not acceptable. We're just can't happen.
So thanks for that. And again, let me just say on behalf, I know of the ARM membership, and I'm sure all the folks who are tuning in to the conference today, that we really want to extend our gratitude to you and your colleagues for the work. Right, we have robust conversations sometimes about this and that, and I think that will continue to be. But we really appreciate the unbiased approach that you take to things. I did want to ask you about some of the recent decisions that the agency has taken in our space. Clearly, the BioMarin decision, most significantly, and the CRL they received, but also the Sarepta decision have been noticed in the community. And I wanted to just ask you if you could articulate what message, if any, the community should take from, let's start with the BioMarin decision, and whether it signals anything different about the way, for example, the agency is thinking about standards for durability.
Yeah, so I am limited to what I can speak about a specific company, and I can't speak about a specific company. But let me just take it back a notch to what I can speak about, which is that nothing is changed at all fundamentally about our commitment to a very strong, enduring commitment to really moving ahead gene therapy here, and to trying to be as flexible as we can be. I do think, though, as we back up to thinking about gene therapy, again, not around a specific company, but around what there is on a given disease, and we addressing a disease with gene therapy. Let's think about that. For a disease for which there's nothing else out there, or maybe there's one thing that's not such a great therapy out there, we're going to be thinking differently about that than we're going to be thinking about a disease for which there were multiple approved, acceptable, and quite effective therapies. And at the end of the day, what one's dealing with, unfortunately, whether we like it or not, is a difference between what is ultimately convenience, right? It is convenience when you have multiple approved therapies, potentially cost issues, versus life or death. And there it's a different benefit risk consideration. And so nothing's changed there. I think for those who don't know, I'm a hematologist oncologist by training. I ran a hemophilia treatment center twice in my life, once at Yale and once in the Harvard system. So I know there's nothing more than I'd like to see than the hemophilia gene therapies come along. But it's very important that when they come along, we understand them, and we understand the benefit risks there. So I can't speak to an individual company, but at least I hope that gives you some insight into where this comes. If it would be the same thing if somebody had a gene therapy that they were addressing a very common disease for which there is an effective therapy or multiple effective therapies, you would want to make sure that you really understood the benefit risk in a different way than you would for any of the mucopolysaccharidoses for which there is not a good replacement therapy, for instance, because there you're dealing with a different benefit risk consideration.
So can you say anything more about, as you think about benefit risk though, does it mean that in a case where there are existing acceptable therapies, that the emphasis will be on a benefit that is on the durability of benefit, or will there be an emphasis on other aspects of benefit as you make that trade-off, or is that always going to be a case-by-case decision?
I think it's going to be somewhat of a case-by-case decision, and I think there's probably some combination, right? It's in some cases, it's so you could imagine you get some benefit of being normal per se, perhaps, or maybe you don't, right? But if you do get a benefit of being normal, how long does that benefit last over time, right? On the other hand, there could be other, it could be a different situation where the issue of not being, so if being normal helps you just be more convenient for a period of time, then it's a different thing than if being normal helps you actually be able to function, right? If being, the difference between a gene therapy goes from not being able to walk from your bed to the bathroom, and then you get it, the gene therapy, and you can, the difference there, the duration may be a little less of an issue, perhaps, when you think about it, than the former case where it's just mainly a convenience factor. And I don't, by the way, I just, for the hemophilia community, I don't, by any means, I don't mean to downplay that it's really not fun to have to, especially in smaller children, to have to deal with putting a needle in a child. But especially with the bispecific monoclonals that are out there now, or the bispecific monoclonal that's out there, there's just a different, in that area, things have changed a little bit over time.
You know, as you know, another part of the conversation that's come out of the recent decisions has been a conversation around communication. And you mentioned, in your opening remarks, of course, that COVID has put demands on the agency. So can you describe, maybe the question is, what's a realistic expectation then for a sponsor about the level of communication? I mean, I think companies would rather have a realistic picture than not, even if it's not the picture they would design if it was completely under their control. So can you just try to characterize that as best you can, given what you're coping with now?
Yeah, no, this is a great question. And I want to just back it up to preface this by saying that our Office of Tissues and Advanced Therapies has a group of people that are incredibly committed to doing as much as they can, and they are working beyond hard. So I think we need to preface what I'm about to say next as this is not to impugn that anybody is doing anything less than all that can be done. They're working way above. If you look at the number of hours, and we do have full-time reporting now, if you look at the number of hours they're spending at work, it's remarkable. Okay, they're trying to do their best. The problem is that what happened here over the past 5 years is something that maybe it could have been predicted, maybe it couldn't have been. There has been an onslaught where you have an office that should have doubled in size, and it's only, it's modestly larger, 20% larger in size, where it really needs to be twice the size it was. And what that leaves us with is a situation that even before COVID, let's just back up before COVID. If I would have had this conversation with you, I would say, I would have said, I'm really not satisfied with the kind of dialogue that we can have with companies, because I think that particularly early on, it's just such a wonderful time for FDA to be able to interact with companies. Once it gets closer to licensure, we have to be much more formal, I think, because there is that more formal negotiation that goes on, and things get more, the stakes are higher. But especially early on, we should be able to have this dialogue that really facilitates setting things up well, so that our knowledge of the entire field, we help leverage that for every sponsor. But we've been so strapped in terms of personnel that it's hard to do that. And so even before COVID-19, I think we weren't doing right by timing, and I think, unfortunately, there probably were some who got worse timing than others in terms of sponsors, just by luck of the draw. You know, there's always a bell curve, and I feel badly for people who are on the longer end of that bell curve. Now, come along COVID-19, and I think it's been really exacerbated further, because we've had the number of gene therapy applications hasn't fallen off dramatically. I mean, some of the trials may not be moving as quickly, but the applications keep coming in. People keep having interest in this area. And the office has more demands on it, because not only does it have those applications, we have COVID-19 related products, stem cell products, exosomes, a whole variety of different things coming in that we have to address, and those tend to take some priority. So that timing has even gotten even somewhat further drawn out. And you can imagine, if you have a workforce that was somewhat tired and overworked to begin with, and they were, that now with this additional load, it's probably, it's led to what I think longer timelines than anyone would like to see. I think companies need to expect that we're doing our best, but the timelines that we have, I don't consider them to be something I'm proud of particularly. We're doing our best to try to fix this problem. I think we're committed as we move now, hopefully as we move out of COVID, as we move into the next user fee cycle, we'll actually have and ask for the kind of resources we need to make this work.
You and I once had a conversation in the midst of or around the time of the government shutdown, about your worries about what that was going to mean for recruiting. And as you said, you'd had a lot of open positions even at that time, anticipating what was going to be this coming wave of applications. You know, then kind of COVID hits, and then for a period, maybe it looked like having a job in the government was a more secure position again. I guess what I'm trying to ask you is, how is recruiting going? I mean, you do clearly have a lot of roles to fill, and I'm wondering just the trend line. How is it going, and is there something that the community watching can do to help you?
Yeah, I'd say that the community watching, if they have people who are interested in it, there are people, I'm sure, in your organizations who are very idealistic. Maybe they're not as business oriented. That's where FDA, we're a great place to work. We're a great place for anyone to spend some time, I think, who's interested. We've done not so badly in COVID-19, in part because, at least on the physicians end and in some of the companies, have not been quite hiring quite as quickly. On the physician end, there are physicians who would prefer to be in a non-practice environment at this time. So I was worried it was going to be worse. We are hiring. We do continue to hire. Some of the issues, just to be frank, we still are not good at getting out of our own way in terms of our hiring processes at the agency. And there's a concerted effort to try to work on that. And I hope that continues, because unless we get facile with, I think we lose good people, because good people don't want to wait months and months before they know that they're going to be able to come someplace. I think they want to have something decided sooner, and so we need to work towards that. I think to the extent that people want to do anything, I think getting the word out that, we welcome people at FDA, whether they want to come here for their life's work or whether they want to just come spend a few years and see what we do and learn, because you do get an incredible exposure. I mean, for me, again, I'm going to come back to this, what keeps me excited about getting rid of COVID is I can't wait to spend more time again with, looking at applications. Because even at my level, I love seeing the breakthrough applications that come in where people are making 5, 6, 7 genome edits to make these really cool T cells that have it's almost like computer logic boards. That is the kind of the excitement that we have. Now, we can't go out and talk about that. And that was just a hypothetical example I gave you, of course, with no relationship to what anyone is actually doing necessarily. But we can't talk about these things, but the experience you get. And then we do talk about them to sponsors in a different way, which is we take that collective knowledge process it, and although we don't speak about a specific application, we try to help sponsors with that knowledge that we gain of the whole field, which in these very rapidly advancing areas is really important. And actually, one of the things that insights I have had over the past couple years is that I continue to believe now that our kind of research reviewer model is actually a pretty good one, especially in this gene therapy space, because being able to be right at the edge of the science is really important. So we are continuing to look for people with that scientific background. So sometimes I may lapse into talking about medical officers, but it's not just medical officers. It's the biologists, other scientists that come in that do incredible jobs as research reviewers, both for chemistry, manufacturing, controls, pharmacology, other things.
Yeah. You've talked in the past about speaking about the kind of scientific component of reviews, that the cycle times for gene and cell therapy may be more akin to sort of devices than traditional pharmaceutical products, and that therefore, you've begun to think about, what are the regulatory practices of the device world that you could apply in the cell and gene therapy world. And you touched at the top of the hour about this notion of kind of review by platform as opposed to product. Could you just drill into that a little bit, talk a little bit more specifically about how you see that getting operationalized, and what else you might draw from the device world for the regulation of cell and gene therapy?
Yeah, so I mean, I think we have to understand that, look, as somebody who worked on small molecules in industry, you develop a process in industry and that same process is used for years, right? Yeah, I guess maybe someone will tweak it. They'll change out a solvent here or there, maybe. And occasionally someone maybe will change one synthetic step, but by and large, that's the process you use. For gene therapies here and for cell therapies, people are always going to be tweaking here. We're going to continue to have advances in how you might be able to better purify, get higher yields of capsids that are fully loaded. So I think we're going to have to figure out how we can advance cycle time such that we don't worry. We're going to have to figure out that we don't worry that we're going to lose activity. I mean, we're going to have to figure out how can we allow products to evolve without worrying terribly that the clinical efficacy of that product is going to disappear. I don't have all the answers there, but I think some of that is going to be trying to understand critical attributes, that as long as that critical attribute is present as you make the change, it's okay to make that change without having to go through a full clinical development program again. It may be that there'll need to be some comparability. You may need to, maybe we'll need to use some real world evidence over a period of time to make sure that nothing is shifted. But I think we have to at least explore how to do that, because otherwise I think we're going to do a disservice here in slowing down the evolution of this. Now, I will say to you, just so that nobody has any illusions, I think our reviewers still, we still have a ways to go to get there, and our reviewers are still concerned when you change processes that you're changing a product. But I do think that we need to start working towards this idea of what changes can we tolerate. And I think one of the first steps of getting there is starting to see, well, if you can change out various vectors, if you can change out various inserts into a vector. So you have a vector that you make by a standard process, and you can start swapping in and out different inserts within perhaps a certain size realm that you can characterize the properties of the insert. Can we find a way that we don't have to go through the whole characterization again? I mean, that to me is that's the simplest case. And then after that, it comes to, are there ways perhaps to change other characteristics of the process in the manufacturing process itself without having to go through and recharacterize not just the manufacturing process, but recharacterize the clinical activity with another study, because you have a different biologic product. So again, questions, but don't have the answers. They're questions, though, that I love thinking about because they will hopefully, if we start to try to answer them and have a dialogue about them, will hopefully help us to leapfrog gene therapy forward in a way that we're not really moving at just yet.
Yeah. You know, you and I have talked about some of these manufacturing challenges over, a couple of years now, and we worked together on ARM launched something called the Standards Coordinating Body, the SCB, to kind of think about where standards could help advance the field. We've been working on a sort of case study best practices guide for gene therapy to try to help folks avoid some of the typical problems that people make in manufacturing and the CMC part of their applications. You mentioned that you guys were also making some grants in the manufacturing space. Can you tell us a little bit more about those and how you guys see you might be willing to, or not willing, but able to help advance the issues like potency assays and comparability and some of just the sort of core challenges that the field is working on?
So we've been very interested in terms of viral vector manufacture. How can one make the process more efficient? And how can one get a better handle on kind of process characterization? What are the critical quality? We want to help get at some of those critical quality attributes. What are the factors that one needs to understand? How can we get better process control so that this becomes something that is able to be used more routinely in a pre-competitive space? We want everyone, this is like, the issue here is that there's a lot of IP that people are developing around this. We don't want IP here. We want something that everybody can use so that they can produce vector efficiently and characterize vector efficiently so that we can see more advances here. And whether that be, and the same goes, although I said for AAV, the same thing goes for lenti production, where it's again also a challenge. So if we're going to make, ultimately, it would really be nice to get to the place where you can start to see enough advances so that the manufacturing becomes kind of like where it is for monoclonals now. Nobody spends a lot of time worrying about how their monoclonal is going to get scaled up anymore. But right now, I think most small companies, they're spending a lot of time figuring out how are they going to get sufficient quantities at a reasonable cost of their gene therapy vector made. And that reasonable cost issue is a very real one because especially as you think about gene therapies where you give on a weight basis to, if you're thinking about addressing something in a larger child, you're getting to needing very large particle numbers that could cost a fair amount of money. So this is a challenge here. So for us, helping in that area, giving out these grants, it's not a lot of money, but in some ways, the nice thing is it at least shows our devotion to this to really, and I think it's helped. We've seen some really good work that we've already seen come along in trying to better characterize the attributes that we need to focus on. So we'll see where that goes. And I know NIH is the FNIH is very interested now as we're moving forward in this individualized gene therapy in thinking about how they can work in this space as well.
Yeah. Are there other core CMC regulatory challenges that are kind of top of mind for you?
Well, I think the one that I would just say, because I hear about it and I can see it being a problem, and I've seen it in COVID-19 even be a problem because of disruption. There's not, it would be nice if there were more standardized reagents that people could just get that were well characterized so that that we didn't have sole source suppliers that go out of business or that run out and again create problems that lead to, every time somebody has to switch a reagent, when you don't we see if you really understand the process, it's easier, right? I think it's if you really understand how to make a thickened sauce, whether you can substitute cornstarch or if you don't have any flour or vice versa. I don't know. And you can thicken the sauce. But if you don't understand, like, I don't know how to do I boil it longer? Do I, add an egg? That might not be good because it'll just congeal, but you just do all sorts of stuff, right? And that's kind of what we do with gene therapy because we don't understand what we're doing sometimes. I don't mean to I feel badly. I'm not trying to insult anyone in the manufacturing area, but what I'm saying is we don't have as good a handle on this. Yeah. So we don't know what to add when, so that when people have to swap out to a new reagent and something doesn't work, what was it? So I think actually that's my other piece of thing that's top of mind is trying to see if we can, I think starting to have, again, this kind of cookbook that you have actually supplies that we can rely upon would be a nice thing as well.
So, we had, you and Dr. Gottlieb made some oft quoted predictions about 10 to 20, gene therapies in 2025, and EMA kind of confirmed that same sense. And, I noticed earlier you used the MIT predictions, not your own. I don't know if that means your own predictions have changed, but we have a little more experience under our belt. So I guess we've had a lot of RMAT designations, but we haven't actually had an RMAT product make it to market yet. And I was on a panel discussion earlier this week where someone said they feel like we're at the point of the sophomore slump, or we're trying to produce, the follow-up hit record, after sort of the first release that put the band on the map. And you can see how that description comes about, right? We've had a little trouble getting kind of out of the gate with some products that we thought were maybe a little closer to market than they've turned out to be. Is there anything other than these manufacturing issues that we've been talking about for the last few minutes that you feel like is kind of causing this, again, not my words, but sophomore slump? And do you feel like it's of a nature that it really changes that prediction that you had made earlier about how many products will be on the market by, say, 2025?
Yeah, I'm not too worried about the number of products that we're going to get to. Could it be on the slower end of things? And maybe it will be. On the other hand, we may make up time very nicely in other ways. I do feel like that manufacturing is a big issue. Understanding the clinical development programs, I think, is another. I would put it to you that I think if we had more time to interact with sponsors, that might be very helpful. In part, I think, because what we have, look, gene therapy, although there are some big players in gene therapy, fundamentally, a lot of gene therapy is done by small investigators, academics, or small companies, and they benefit a lot from interaction with the agency. And to the extent that we could have more of that interaction, hopefully we could avoid, miscommunications or different expectations and that could hopefully lead us faster down the path. I can't promise that, but having more timely communication and perhaps broader communication, I think. How could that be bad, right? It's like, I mean, I don't know. I mean, arguing against that, it's arguing that this is hard to argue against, right? I mean, at the worst, if I'm wrong, we'll have more timely interactions, and that will speed things up. And at the best, some of the things that have led sponsors to perhaps go astray down development paths that haven't been productive, we'd have less of that, because sometimes people just get tired of waiting for us now, and they just kind of go along their way, and that leads then to having to find our ways together again. And so it'd be nicer to have those dialogues sooner rather than later.
Peter, you talked about kind of one of the things that you're sorry has been slowed down a little bit is your efforts around international harmonization. And, I wondered if you'd say a little bit more about what you thought were, kind of the things that you're pushing the hardest on. There's obviously a lot of issues when it comes to international harmonization, and whether you feel like, in addition to COVID, obviously there's a transition about to occur at the EMA, and whether you have any sense yet about the significance of that. Professor Rossi was obviously very passionate about cell and gene as well, and was I think a strong partner to the community. So wondering if you could speak to those issues.
Yeah, so there will be a transition at EMA, there's no doubt. Emer Cook is someone who came from, was at EMA, went to WHO, now going back, and I hope she'll continue in the same vein. There's been an interesting, dealing with Europe is a very interesting, an interesting dynamic, because there's this issue of some of the gene therapies are done locally at hospitals under this hospital exception that the member states have, which does make it a little more challenging for EMA. That said, I don't see a ton of space between us and the EMA here. I do think actually having more dialogue there, so we adjust. Sometimes we're a little off on the manufacturing expectations, and it's kind of interesting to me, because it's not like we need more and they need less. It's that they're just different, and if we could align those, that's really would be much more much more helpful to sponsors, so that they don't end up with two packages that they have to do to satisfy two different regulatory authorities. I get the sense that any number of other regulatory authorities, if they saw the leadership between EMA, PDMA, FDA kind of coming together, a lot of the other regulatory authorities, I think, would just love to come along for the ride. And I think probably figuring out a way that we could help them do that.
Mhm.
And again, they need to be competent regulatory authorities, don't get me wrong, but helping them, making that easier for them would be an important thing. And I think there's also a certain amount of education that's going to be necessary in low and middle income countries so that we educate them so that we don't, at the same time we have gene therapies coming on that are well designed, good, safe, effective therapies, that we don't have kind of a gray market or a black market or whatever kind of market you call it occurring where people go for bargain basement discount gene therapy to regulatory places where the regulatory systems are not developed. So we're trying to do that. That was our goal of this, of working with WHO actually, was to try to do kind of global education so that we don't create niches where less than savory therapies can flourish, because it's unfortunately, as we now have learned all too well with social media, people can go very far astray and based on advertising, etc. We want to make sure that what's out there doesn't discredit what's good. I mean, you can imagine all it would take is one genome editing experiment, so to speak, in a place that doesn't have the appropriate regulatory knowledge to essentially put a chill across genome editing across the globe. So we feel like it's our obligation to make sure that we educate various regulatory authorities. So I'm hoping that we get there. I think they could be very good all around, good for diseases where it would be, rare diseases where it'd be good to get therapies proliferating across the globe. It might make some commercially non-viable diseases suddenly commercially viable, right? Because if you have 50 patients in the US, 75 to 100 patients in the EU, 10 patients in Japan, you go figure, you certainly may have suddenly reached commercial viability in a way you wouldn't otherwise.
It's interesting what you say about the kind of manufacturing requirements. We did a study about kind of disharmonies between EMA and FDA regulation in the field. And the result of it was pretty useful. We've shared it with your team and with the EMA team, but it wasn't like, oh, there's this one thing, if only we could fix this one thing. It was, as you said, it was kind of a little bit of a menu of smallish things, but just kind of in toto meant that there were kind of these two reasonably different packages that had to be put together. So I guess I would just underscore, your sense of it was mirrored by the analytical work that we did. Let me just finish up. You've been generous with your time. With one other question, going back to this sort of device model, right? So the administration recently announced that for certain breakthrough devices, that there's going to be a kind of simultaneous approval by the FDA and a coverage decision by CMS, right? Which is sort of a holy grail for sponsors, of course, to be able to get sort of market approval and at least CMS reimbursement, coverage and reimbursement simultaneously. Do you see any possibility of taking that model from that kind of what seems like a kind of limited case study and applying it to our domain, or if not that model exactly, an opportunity for us to work more closely, kind of in a three-way conversation with the FDA, CMS, and the sponsor in order to shorten the time from approval from you to really having a situation where products can get to patients, and which involves CMS decisions as well.
Yeah, I mean, that's something that was, again, something interesting. We were trying to work on that some before COVID. I think it's an interesting departure point for a conversation, and a good one, because it really has to do with us trying to help CMS, educate CMS as products come through within the confines of what we can do. but I think there's probably some room for progress there. And I think we probably should start with the low-hanging fruit, which is educating them in a manner where we don't violate anything, don't create any problems for them. And then if need be, we can think about other ways of moving forward. But it is something we've thought about. And, you know, especially as you start to think about, you know, gene therapies that might reuse vectors, you'd hopefully be able to find ways to make it so that could move ahead as a kind of a test case in a relatively quick manner. But we'll see. I'm happy to. I think it's a good place to start a discussion here, because again, if you don't help patients with the therapies, why are you developing them in the first place? And the delays, especially for I mean, the thing that's heartbreaking to me every time I hear of someone who's lost a child where they were this close to a, you know, just hairbreadth away from being able to be on a gene therapy trial, it just kind of breaks my heart. So be nice to, we'd be happy to kind of work on that. And I think that may be, unfortunately, something that's also a somewhat post-COVID thing, because there's only so many things we can do during the day and early morning and late evening hours these days.
Yeah. Well, Peter, is there anything else that you can think of? We talked about recruiting, but is there anything else, given that, you know, our membership absolutely shares the FDA's goal of getting these safe and effective therapies to market? Is there any other way that ARM and its members can be supporting that effort right now that you'd like to speak to?
I want to thank you so much. I mean, I think by continuing to do what you're doing, we appreciate it. I think by supporting a strong independent FDA that follows science is an important thing, and we really appreciate that. I think by working together to educate the community, it's very important. So that's not just both the community of sponsors and the community of consumers. So I think it's that to me is what we can ask for here. And I think probably as we move forward, as we move forward into the future, it will be support for the growth that we're going to need to actually really flourish in this area. I mean, I think to me, what I would say is if we want to remain global leaders in the United States of gene therapy, we're going to have to be able to assist our industry in growth in a way that is commensurate with that. And that means that kind of the support for having an adequate number of people to be able to do that is probably an important thing.
Yeah. I think we certainly appreciate that. And as you said, you've got a lot of good people there who are working a lot of long hours, but that alone isn't gonna build the capacity that we need to cope with what's obviously coming out of the pipeline right now.
Yeah, I mean, I would say that the problem here is that, you know, you're most creative when you're, you know, there's if you think about most processes, they work best at about 80 to 85% efficiency, right? You go, once you're working close to 100% or at 110%, you don't have that time to actually contemplate and think about, well, could we have done a little differently? You tend to just work by rote because you just got to get the job done. And to the extent that we were actually not always working right at capacity, or as we have been at 110, 120% or more of capacity, I think we would have more time to kind of contemplate alternatives and have the kinds of more dialogue with sponsors that would allow us to try to be a little more creative in what we do. That's, again, I can't say I have some scientific documentation that that's going to happen. But I just would put it to you that that's my hypothesis on how it would be.
I know you have a lot of heart in the effort on kind of N of 1 gene therapy or the bespoke gene therapy project. And I just want to say to you and to anybody else who's involved in the conference that we are having a panel specifically on that with some of Dr. Marks's colleagues from the FNIH to really talk about how that's going to work and what the current program plan looks like, and if people are interested in becoming partners to that effort, what that would look like. So we didn't talk too much about that here, but there is a chance to learn more about that. So with that, let me thank you again, Dr. Marks, for your time today and your forthright answers to the questions that we've got. And we look forward to working with you more in the future.
Same here. Thanks so much for having me today.
All right. It's a pleasure. Thanks much.